I'm Vincent Racaniello, and I would like to just reminisce, a little remembrance of polio past for those of you who read any Proust, right? I've known Kostya for a long time, and in fact, I even reviewed your program years ago. I don't know if you remember.
I do.
You do remember. So we have almost parallel careers, just at different places. So I thought I'd give a little history and overview of what we have done. Poliovirus has infected humans for thousands of years. We know this because there are sporadic reports of the disease in the literature, this Egyptian carving that you've already seen. But it was not epidemic, a case here, a case there. That changed at the beginning of the 1900s when suddenly the infection became epidemic. It occurred in outbreaks of increasingly large numbers, as you can see here. And eventually it was sorted out that this is because of the development of modern sanitation. Before 1900, the virus circulated freely. Every infant was infected shortly after birth. Feces were everywhere. And then, of course, in 1900, the greatest invention of humanity, the toilet and sewers, removed fecal material from the early years of life. The early years of life, of course, being protected by maternal antibodies, so babies would get infected, not develop paralysis. And then once the toilets are developed, we delay infection until there are now thousands and thousands of individuals who are now susceptible to infection. We have outbreaks of polio. So we call polio a disease of modern sanitation. I love to point this out and say, had we had genome sequencing in 1908, we would have blamed the emergence of epidemic polio on a mutation that we saw in the genome. Virus isolated in 1908 by Karl Landsteiner, who injected sterile filtrates from the spinal cord of a boy who had polio. The monkeys developed lesions of polio. Remember, this is the beginnings of our understanding of what a virus is. They were only discovered at the end of the 1800s. But he put it through a filter that passed viruses and not bacteria. I also would like to point out early misdirection by so-called experts. Simon Flexler, the first director of the Rockefeller Institute for Medical Research, he started to work on polio. As soon as it was discovered, he jumped in. He thought that the disease in monkeys was the same as in humans. He thought the virus is strictly neurotropic, entered the nose, went directly to the brain and spinal cord. Of course, he worked with a strain that had become neurotropic via endless monkey-to-monkey passages because we didn't have freezers to store our stocks in. We stored them in animals. And so he wrote a press release in 1911. A cure is about to be rewarded within 6 months, according to Dr. Flexner. A specific remedy may be expected. We have discovered how to prevent the disease, and the achievement of a cure, I may conservatively say, is now not far away. His cure was a nose spray, which coated the olfactory mucosa with aluminum, and it worked in monkeys, but failed in 10,000 children. Now, I went to do a postdoc with David Baltimore in 1979. I had finished a PhD with Peter Palese. And this was an important time because the moratorium on cloning viral genomes had just been lifted. In the late 70s, recombinant DNA technology was developed, and scientists thought maybe it's dangerous and we should pause our work. And so the Asilomar conference was convened. And during that time, you could not clone more than 2/3 of any viral genome. But when I went to David's lab, that moratorium had been lifted. So David said to me, I want you to clone the genome, do its sequence, and see if you can get an infectious DNA. And I said, sure, in all my naivete, right? Little did I know. Well, there, of course, is the subject of much of today's talks. We did do the genome sequence. This was the day when you could publish a genome sequence. Eckhard Wimmer's group had published it just a few weeks before. This took me a year to do by Maxim and Gilbert. Nowadays, you could do it in a half an hour. And I think the lesson is very clear. Back then, we worked on one virus, pretty much. and that was all you could do. Now you can work on thousands, tens of thousands of genomes, of course, with high throughput sequencing. So I accomplished that part of his missive, and we, of course, saw the open reading frame and the non-coding regions, which many people have since worked on. Then that was achieved by sequencing a DNA copy of the genome made by reverse transcription, cloning into a plasmid. He then said, all right, Vincent, take the plasmid, just put it in cells and see if it's infectious. I said, no, don't I have to put a promoter in there? He said, no, just throw it in cells. So I did. I took the plasmid with polio DNA, put it in cells, and out popped, on the very first experiment, infectious poliovirus. The moral of that is just do the simplest experiment. Don't get fancy unless the first try doesn't work. So I took that plasmid to Columbia, where I started my lab in 1982. And I wanted to make a mouse model for studying disease. That's what I was really interested in. And at the time, there were mouse-adapted variants of poliovirus. One of them is the Lansing strain here, which was derived by Charles Armstrong in 1939 by multiple passages of the Lansing strain in mice. So this virus, when intracerebrally inoculated into mice, the mice get paralyzed. The virus replicates only in the brain and spinal cord. You can't infect any mouse cell line with Lansing. We always wondered what the receptor was, and later I'll get back to that. Then in some work we did with Eckard, we found that substituting a 8 amino acid sequence in the VP1BC loop of type 1 Mahoney, which does not infect mice. So here's the viral particle. In white is the VP1BC loop. If you take that piece out of Mahoney and put in the Lansing sequence, that virus can now infect mice. It paralyzes mice. We call that virus 414. So we could narrow it down to that small region. And remember, the receptor is binding in a canyon around that 5-fold axis. So somehow this is influencing interaction with a mouse receptor. Now, we fast forward many years later. Amy Rosenfeld is making brain slice cultures for other reasons. I think this was enterovirus D68, most likely. So these are day 10 mouse brain slice cultures. And she said, I'm going to throw Lansing in and see what happens. And lo and behold, Lansing replicates in these brain slice cultures. They are replicating in astrocytes. Lansing is replicating in astrocytes. This is a stain for GFAP. This is a stain for a viral protein. And the merge is here. Astrocytes. Who would have guessed? This is supposed to be a neuronotropic virus. So in there is the receptor for Lansing. You can use 414, which is Mahoney with the Lansing BC loop. And if you do it quickly, maybe you can find the receptor. Otherwise, you have to go to a polio sensor facility. But I think it would be very interesting to understand what that receptor is. After working with that virus for a number of years, we made a transgenic mouse model. Kathy Mendelson had identified the polio receptor in my lab, along with a collaboration with Eckhard Wimmer again. Rebal Ren, another graduate student, made transgenic mice, producing that receptor. You've heard about them. They were the first animal model, transgenic animal model for a virus disease. These animals become paralyzed after inoculation of the virus. Interestingly, they do not become paralyzed after being fed the virus orally. The gut of the mouse is not the same as the human gut. It's protected by interferon, because if you knock out the interferon response, then the gut can be infected. But who wants to use immunocompromised mice to study pathogenesis? Now, as you've also heard today, the development of genome sequencing allowed many laboratories to understand the determinants of attenuation of the Sabin strains. Bless you. And here they are. And it was shocking to see so few changes in vaccine strains, which I would say today would preclude them from being approved as human vaccines. And of course, all of them have a change in the 5' non-coding region. Now, we worked on this in our mouse models for a number of years. Here they are in domain 5 of the 5' non-coding region. The 3 OPV serotypes, 1, 2, and 3, have very specific changes. For example, type 3, a U in the vaccine and a C in the wild type. And I think this is a wonderful example of how a virulence determinant need not be a protein coding gene. It can be a non-coding gene. So I use this for teaching all the time. We introduced these changes into the Lansing strain, as Jeff Allman mentioned this morning. We have two Lansing viruses that differ only by one base at 472, either a U or the C. The U from the vaccine strain, the C from the wild-type parent. And the C-containing viruses, the LD50 or the PD50 is 9,000 PFU. The U virus never paralyzes mice, no matter how much you put in. The C virus grows very well. This is PFU per gram over days post-infection, and the U virus is cleared. And so that demonstrates the dramatic effect of a single base change in a non-coding region. Now, Phil Miner, very early on, after his first child was born, David Miner, he collected his son's diapers after the son got the first dose of OPV and brought them to the lab, sequenced them for type 3 polio, and found that within 35 to 48 hours after immunization, that U at 472 rapidly reverts to a C, or I should say viruses with a C are rapidly selected for their greater fitness. And this happens in almost every child, probably every child that receives OPV. And the lesion score of those viruses in a monkey neurovirulence test is quite high. It would not pass, it would not be released as a vaccine, obviously. So this occurs in every child. And we've always been interested in how that one base change attenuates neurovirulence. And we published this paper a number of years ago in the JCI that said the effect of the 472 change is something after internal ribosome entry, because we made a reporter adenovirus with the 5' UTR of poliovirus type 3 with either a C or a U, and we measured the reporter or the iris activity in different tissues of mice, and you can see that the wild type 5' UTR has a very high activity, and then the U containing virus, or I should say 5' UTR, attenuates translation equally in different tissues. So this is not a CNS specific effect. It reduces translation in most tissues. And so that's part of the lack of fitness of this particular virus. We also did a series of inoculations in transgenic PVR mice, and I want to point out one to you, which is important, because here's P3 Sabin with a U at 472. If you inoculate intracerebrally into 4-week-old transgenic mice, there is no paralysis. But if you use instead 1 to 2 day old mice, the LD50 is about 60,000 PFU. So the host also matters. It's not just the base at 472. In the case of a 1 to 2 day old mice, they have no immune system, neither innate nor adaptive. And so that allows even a poorly fit virus that has a U at 472 to multiply. And so I think that tells us that the kids who get VAP have some immune defect, most likely in the innate immune system. As you have heard and all know, we have in 1988 a declaration to eradicate poliomyelitis initially, and then somehow becomes poliovirus. So can you actually eradicate a viral disease? Well, we did smallpox, the only major human disease to be eradicated. And D.A. Henderson, in his book, D.A. Henderson, of course, was the architect of the eradication program. He said there are 3 features of a virus that are essential for eradication. First, the replication must occur only in one host. Okay, check for poliovirus, humans only. Vaccination must induce lifelong immunity. Okay, I'll give you a check on that one, all right? The third one, asymptomatic infections do not contribute to viral spread. That's the case for smallpox. When you see a kid with a rash, that's the kid who has smallpox. Nobody else around him or her has smallpox. But when you see a child with AFP, there are lots of infections around that child that are shedding virus. According to D.A. Henderson, and I asked him this in an interview, can we eradicate polio? He said, nope. And so if you don't believe me, you can believe D.A. Henderson. So if you just look for paralysis, you're not going to eradicate. And you can't look for virus in every wastewater treatment plant or environmental source in the world. So, so even saying type 1 is eradicated may be wrong because we don't look everywhere. Let me end on some other enteroviruses. As you know, look at this Picornaviridae family. When I started working on this, there were 4 genera. Remember, Anne? 4 genera. Now there are 40, and most of them don't have infectious virus isolates, they're sequences. But anyway, here's the enterovirus genus, and here we have polioviruses, enteroviruses. The rhinoviruses are down here. And all these other enteroviruses in bold, those can also cause AFM, acute flaccid myelitis. And we got interested, when Amy returned to the lab after her PhD, a number of years before, she got interested in enterovirus 68, which was isolated in the 60s from children with pneumonia. Very few infections detected until 2014, when, as you know, there was a big outbreak throughout the US and Canada, and a number of these were associated with paralysis. And CDC has tracked these AFM cases associated with EVD68s, and there have been surges every 2 years, presumably reflecting the buildup of susceptible children without immunity. And then, of course, the pandemic put a damper on that, and we're just starting to see some cases return. So the fact that this virus has been around since 1962 and yet did not cause paralysis until, at least as far as we know, until 2014 is a very interesting question. So Amy asked the question, if you take a series of isolates from the 60s to the present, can they infect human neurons? Are they neurotropic? And the answer is yes. This virus has been neurotropic since the 60s. It has not acquired neurotropism. It's always been there. We don't know why it's causing paralysis in children now, but it probably will take some sleuthing beyond just looking at the sequence, just like poliomyelitis at the turn of the 1900s. Not only does it infect neurons, it infects astrocytes as well. And you may be asking, well, what does that have to do with the pathogenesis? When an astrocyte is activated, in this case by infection, it secretes factors that can damage neurons. So this is could clearly play a role in the pathogenesis of paralytic disease. So Amy has taken that work with her here to at the FDA. Now, a few last comments on viruses in the central nervous system. Many viruses invade the CNS: poliovirus, Zika virus, EV68, West Nile, measles. For the viruses we have looked at, EV68 and also Zika virus, similar story, 1947 to 2015, no paralysis or no neurological disease caused by Zika virus. People are saying, well, it must have mutated. No, actually, in 47, it already had the ability to reproduce in the CNS. All of these viruses are efficiently transmitted by shedding, either from the gut or the respiratory tract or via mosquitoes. The CNS invasion is a dead end. It is not selected for directly because in humans anyway, once the virus gets in, it stays there. And transmission to another host, of course, is the biggest selection factor for viruses, because if they don't get to another host, that's the end of the line for them. So it's not selected for. It may be an accident of some other property that's selected for, but in itself, it is not selected for. Finally, last story, again, the last set of experiments that I actually did at Columbia along with Amy, enteroviruses are genetically very similar, and it's been reported in the past that humans here have cross-reactive activities among different enteroviruses. So we asked a very simple question: are there cross-species anti-enterovirus neutralizing antibodies? We immunized mice with different enteroviruses and did cross-species neutralization assays, and the answer is yes. We can have antibodies against poliovirus that neutralize EVD68 and vice versa. In fact, antibodies against poliovirus that neutralize rhinoviruses. So the contribution of that is potentially very important, probably beyond what we think, and that'll be one of the things that Amy is working on in her lab. I have to end with this wonderful letter. I have a bunch of letters from Albert Sabin that he sent. This is one from Olen Kew at the top, and the bottom, the handwritten one, he sent to me. And this first one, David Baltimore and I had published this paper saying, oh, we have this infectious DNA. We can make polioviruses that do not revert to neurovirulence. Well, I guess we can't, given our more recent experiences. But here's what he wrote to Olen Kew. In my judgment, molecular biologists are limited in their knowledge and understanding of the large number of biological properties of polioviruses which have been measured quantitatively. Each property is not an all or none manifestation, but varies not only in mixed populations, but also in the progeny of single virus particles. He already knew what a quasi-species was before it was discovered. And please, virulence and neurovirulence are not single biological properties. The limitation is not in what can be tested, but in the laborious nature of some of the important tests for which the excellent molecular biologists are neither trained nor inclined to become involved in. Accordingly, they seek easy ways out that are based on untested and therefore doubtful assumptions. All right, so you get the picture. He didn't really like me to write, make viruses that can't revert. And then the one on the bottom here, from 1991, I had been in my own lab for a while, and I used to send him our papers. And he would write back and say, you either have read none of my papers, or if you did, you ignored them, because none of your papers reflect that. And then he closed one of his letters with, enclosed are copies of recent correspondence with Roisman to show that you are not the only distinguished molecular biologist with whom I have problems. I'll be 85 next month and don't expect to be around much longer. And it's the 30th anniversary of his passing what, this year, right?
Yes, March.
All right. So I wish he were around today, not only for polio, but for SARS-CoV-2 and a lot of the fiascos that go on with that virus, because he had even though he lacked insight into certain things, he didn't believe that reversion was associated with many of these polio cases. He still had such insight into pathogenesis that few people have today. It's really tremendous. Anyway, so Amy Rosenfeld was my last trainee. Last year she left to come here to the FDA, so I closed my laboratory, and I focus now on communicating virology and other sciences as well. And so you could, if you're interested, you could go to MicrobeTV and check them out. And I thank you for your attention.
Okay, so what I'm going to try and do in the next 15 minutes or so is summarize the last 70 years of polio vaccine research in about 15 minutes. So in 1952 alone, there were 58,000 cases of polio that were reported in the United States. 65% of cases occurred in children between 5 and 9 years of age. 90% of children and 100% of adults living with someone with polio became infected. Arguably, there was no disease that was more feared. So one of the first to step forward to make a vaccine was Jonas Salk. I mean, he had trained in the laboratory of Thomas Francis at the University of Michigan. He watched Thomas Francis take influenza virus, grow it in eggs, purify it, and inactivate it with a chemical, and that's basically what he did. He grew polio virus up in monkey cells, monkey kidney cells. He then purified it and treated it with formaldehyde at a certain pH, at a certain temperature, and found that he could get roughly a million-fold reduction in infectious virus over a 3-day period. He then assumed that if he treated for an additional 3 days, he would get another million-fold reduction, and an additional more 3 days, another million-fold reduction, or essentially a trillion-fold reduction. You can measure the area above the x-axis, but the area below the x-axis you're not going to measure. You're not going to see whether you have one infectious particle per million mLs. You have to assume that's true. That's the faith part of this graph. And so what then happened was arguably the largest trial of a medical product in history. This is what children looked like in the 1950s. 3 doses of vaccine were given to 420,000 children made by 2 veteran vaccine makers, Eli Lilly and Park Davis. 200,000 children were inoculated with placebo, and 1.2 million children served as observed uninoculated controls, which was really a concession to Dr. Salk, who did not want to vaccinate first and second graders in the 1950s and the mid-1950s with placebo. He just couldn't conscience that. Efficacy was 65% against type 1, 100% against type 2, and 96% against type 3 induced polio. And Salk's vaccine was then released on April 12, 1955 by these 5 companies that had basically this number of doses distributed. 2 weeks later, this headline appeared in the New York Times. One firm's vaccine barred 6 polio cases were studied. This was the beginning of what eventually was called the Qatar incident. There were 120,000 children who were inoculated with live, fully virulent poliovirus as Qatar failed to fully inactivate that vaccine. 100,000 family and community contacts were also infected as basically we started a large man-made polio epidemic. At least 40,000 children developed abortive or short-lived polio, 164 were permanently paralyzed, and 10 were killed in what was arguably the worst biological disaster in this country's history. Of interest, Qatar wasn't the only company that had a problem inactivating poliovirus. Wyeth also had a problem. This was an investigation done by Neil Nathanson when he was an EIS officer at CDC. And what he found also was that Wyeth, which made vaccines for children in the Northeast, also had a problem in activating the vaccine. Frankly, all 5 companies had difficulties in activating the vaccine. It probably should have been more fairly called the scale-up incident, because the line, at least in the hands, especially of Qatar, didn't remain straight. So with improvement in safety tests and filtration occurred as a consequence of this. It was really in many ways the birth of vaccine regulation. Within 1 month of the incident, the Laboratory of Biologics Control was changed to the Division of Biologic Standards, as Dr. Marks alluded to earlier, and the number of full-time professional regulators increased from 10 to 150. This was actually the birth of the term consistency lots. And so then we stepped back into the game after about a 2-month hiatus. Jonas Salk's polio vaccine was back on the market. And as you can see, between 1955 and 1962, there was a dramatic decrease in the number of cases of paralysis caused by poliovirus. But that vaccine then was ultimately replaced by Albert Sabin's vaccine, who you've seen here in one of those rare moments when they're talking amicably. And Albert Sabin had trained in the laboratory of Max Tyler at the Rockefeller Institute. Max Tyler had created the live attenuated yellow fever vaccine by serial passage in non-human cells. So that's what Albert Sabin did. He attenuated poliovirus 1, 2, and 3 by serial passage in monkey kidney cells and monkey testicular cells. His trivalent live vaccine then was introduced in 1963 and replaced Salk's vaccine. Because Sabin's vaccine was shed in the feces, about 25% of people in the home of someone given the vaccine seroconverted, so-called contact immunity, which was viewed as valuable. Sabin's oral vaccine also induced intestinal immunity, as was stated earlier. Sabin also argued that only his vaccine could induce long-term immunity, so-called convalescent immunity. He argued that that was a consequence of natural infection or a consequence of a vaccination with a live attenuated vaccine, but that if you gave an inactivated vaccine, that would require more frequent booster dosing as you got older, which was not true. And I think Salk, frankly, had showed that his vaccine could induce an anamnestic response. So, but nonetheless, that was his notion. And obviously, his vaccine was cheaper since it didn't require paramedical personnel. It wasn't a shot. It was just given as drops initially on a sugar cube and then ultimately just drops in the mouth. And so because of this, because of Albert Sabin's vaccine, we were able to eliminate wild-type poliovirus from the United States by 1979. But there was a problem with Albert Sabin's vaccine, which is that it could cause vaccine-associated paralytic polio. The live attenuated strains of the vaccine could revert to neurovirulent type, so-called vaccine-derived poliovirus. Estimates of this VAP range from 1 in 750,000 for the first dose of OPV to 2.4 million for all 3 doses. Because Sabin's vaccine was certainly attenuated for growth in the central nervous system, but it wasn't attenuated for growth in the intestine, and the virus could reproduce itself for weeks and sometimes months and evolve. And although the vaccine virus rarely reverted to neurovirulent type, the fact of the matter is that it could. So although by the late 1970s would eliminated wild-type polio from this country, we hadn't eliminated polio because every year there would be 8 to 10 cases of polio caused by Albert Sabin's polio vaccine. So to eliminate VAP, in January 1997, the Advisory Committee of Immunization Practice recommended that we move to a sequential schedule initially, 2 doses of IPV followed by 2 doses of OPV. I actually came onto the ACIP in 1998, and John Modlin, who was the head of the ACIP at the time, asked me to head this working group. My goal was to eliminate OPV entirely and move to an all IPV schedule. But surprisingly, there was pushback just in general, even about moving to that sequential schedule. One was the notion by one of our committee members that we would be spending about $4 million per case of VAP prevented, and that, at least to this person, was not a best use of funds. The other, surprisingly, that there were 2 senior members of that committee who still did not trust that straight line theory of inactivation following the Cutter incident. They feared that the inactivated vaccine still could contain live poliovirus. So that was also part of the pushback. But in any case, we were able to move away from the oral vaccine. I asked John Salamone, who was a founder of a group called Informed Parents Against Vaccine-Associated Paralytic Polio. His son had been paralyzed by the oral polio vaccine, and he would go to American Academy of Pediatrics meetings, and he would have his booth. It wasn't treated particularly well, but he was a true vaccine safety activist because he really had a vaccine safety issue. He was very respectful, and ultimately the CDC gave him an award for his advocacy as we moved to a fully inactivated polio vaccine schedule by the year 2000. And with that, we eliminated VAP from the United States, at least until June of 2022, when there was a case in Rockland County of a 27-year-old man who visited an emergency department in New York following 5 days of low-grade fever, neck stiffness, back and abdominal pain, constipation, and lower extremity weakness. He was hospitalized for 16 days with flaccid weakness of his lower extremities. As part of the National Acute Flaccid Myelitis Surveillance Program, the case was reported to the New York State Department of Health and ultimately the CDC. RT-PCR and sequencing of stool specimen by the New York State Department of Health laboratory identified polio type 2. Additional sequencing identified the virus as a circulating vaccine-derived type 2 strain that differed from the Sabin oral polio vaccine strain by 10 nucleotides in one of the viral proteins, suggesting transmission of the virus for at least 1 year. Roughly, vaccine-derived paralytic type 2 causes paralysis in 1 per 2,000 people, meaning that this man was probably the tip of a much larger iceberg. And indeed, in this publication in Morbidity and Mortality Weekly Report by the CDC, vaccine-derived type 2 paralytic polio virus was detected in 69 separate wastewater samples in Rockland County and in neighboring Orange County, New York. So, according to the New York State Immunization Information System, statewide immunization for 3 doses of polio vaccine among children less than 2 years of age was 79%. In Rockland County, the immunization rates were 60%. But in the zip code where this man lived, who had been paralyzed by CDVPV2, immunization rates were only 37%, which explains why we saw what we saw. So, once again, people were scared of polio. On July 21st, 2022, the Rockland County Health Department issued a health alert encouraging those who were unprotected to be vaccinated. And indeed, about 5,000 polio vaccines were administered in that county since that confirmed case. So, which brings us to this question, at least for the United States: how can we stop the spread of this circulating vaccine-derived polio type 2? So you could argue the best strategy would be a vaccine that induces intestinal immunity but doesn't revert to neurovirulent type. So the collaboration between scientists at the University of California, San Francisco, the National Institutes of Biological Standards and Control in the UK, the US FDA, which is why we're here today, and the CDC created a novel OPV type 2 strain, NOPV2. And preclinical trials showed that NOPV was immunogenic and stable. This is a paper by David Ye, and he's I think sitting in the audience, and Dr. Andino is the senior author here, showing what has already been extensively described: that a strain was created that is more stable, that is less likely to revert, and also less likely to recombine with circulating enteroviruses. So the WHO approved NOPV on November 13, 2020. Since March 2021, about 600 million doses of NOPV have been distributed in 28 countries to combat outbreaks of CDVPV2 in Nigeria, Liberia, Benin, Congo, Tajikistan, Sierra Leone, and Niger. At least 38 countries, I guess now 39, have now met the verification requirements for administration of NOPV. So should NOPV be used to quell outbreaks of this virus, CVD-PV2, in the United States? So here was a paper that was published in the British Medical Journal with the headline, "The U.S. needs to prepare to introduce the novel oral polio vaccine," with this statement. In late July, the New York State Department of Health reported that a person had been paralyzed as a result of a polio infection likely acquired in the United States. Subsequently, the same variant of poliovirus was detected in sewage samples in several counties in New York, indicating that hundreds of thousands of people may have been recently infected. On September 13, 2022, the United States was added to a list compiled by the WHO of countries with circulating poliovirus. Quote, New York needs to be prepared to introduce NOPV. So the concerns here are those that have been expressed already. We need to determine the extent of the problem by testing wastewater samples in all major U.S. cities, and the CDC needs to do this. NOPV is a replicating virus. It is a live virus, and therefore it always had the capacity to evolve. IPV will not stop the spread of CVD-PV2, but it will prevent paralysis. Therefore, a highly vaccinated population with IPV solves the problem seen in New York. Do we really want to reintroduce a live attenuated polio vaccine in the United States? And then to get to a point that Dr. Andino already made, I mean, as was said previously, and as you would know, for anybody who watches TWIV, this vaccine can evolve. It's still a live virus and always had the capacity to revert to neurovirulent type. And that's what happened. There were 6 children in the Democratic Republic of the Congo and 1 in Burundi that were paralyzed by a revertant type 2 strain contained in NOPV. We are disappointed, said Ananda Bandyopadhyay, Deputy Director for Technology Research and Analytics for the polio team of the Bill and Melinda Gates Foundation. Any such outbreak is disappointing. At this time, around 600 million doses of NOPV had been administered in 28 countries. So I'll stop there. I guess as someone who'd been on the ACIP during our switch to IPV, it took a lot for us to not use a live attenuated poliovirus strain in this country. So I think for me, it would be a defeat if we went back to a live attenuated strain when we can solve the problem with a highly vaccinated population with IPV. But I'll stop there, and thanks for your attention.
I worked at NIBSC, which was a sort of regulatory agency, if you like. So the line that I'm going to take is really to do with the regulation of vaccines and how you make sure that they're good and clean and safe, right? And Kostya's big contribution to this, and I chose this title because it's big and explicit, and I won't forget what I'm supposed to be talking about. Kostya's big contribution, really, to my mind, is MapRec for type 3, because it introduces the possibility of a molecular test to replace a nasty and unpleasant and complicated animal test. And I think it has implications beyond polio and beyond MapRec, as a matter of fact. So what you can see is this is a logarithmic scale on the left-hand side. And what you can see is that in the first instance, in the 1950s, the incidence was about 10 cases per 100,000 head of population. And on a logarithmic scale, by the time you get to about 1980 or thereabouts, this is the result of the vaccines. Okay, so the Salk vaccine in 1955 and the Sabin vaccine in the 1960s in particular. And you can see that it's gone down by 3 orders of magnitude, which seems like an awful lot. But there's a sort of slight grumbling number of cases left over at the end of it. And if you put this on an arithmetic scale, you can see that by around the 1970s, polio really was not much of a problem, okay? And clinical people really didn't think too much of polio as an area of work because it was all gone. Okay? This was not, in fact, the case. And this is where I start. So I'm starting not at the beginning, but in the middle. Okay? What was going on there was that while there weren't that many cases in either the United States or Great Britain, nonetheless, there were lots and lots of cases outside. The vaccine was making no dent whatsoever on cases in the wider world, especially in the tropics. Okay, so the possibility of importation was always there. So you really had to keep vaccinating no matter what you wanted to do. Okay, so vaccine programs had to continue in developed countries. Okay, the other thing is that residual number down the bottom was a sort of grumbling number of cases where it wasn't totally clear what it was about. So not much of an issue. Okay, clinicians didn't really think much of it. But you still had to keep vaccinating because of the possibility of importations. Okay. In addition, the residual cases that you see down the bottom there, many people in the field did believe that they were caused by the vaccine. This was not a view which Albert Sabin shared, and he didn't share it very, very loudly and vociferously, though anybody who knew Albert Sabin would know how strong he felt about this. However, it was a convenient fiction from the vaccine givers that you couldn't tell polio strains apart. In other words, you couldn't say whether this was a wild type leak or a vaccine reversion. Okay, but most people, I think, thought that you were looking at vaccine reversion. And very smart academic people who knew everything talked about the poliovirus, and what they meant by that was the Mahoney strain. The strains were not to be told apart. Okay. But that did mean you had to test the vaccine batches to the best of your ability, and that meant monkeys, which is a disgusting and very expensive test. So polio had a study group. Sabin had given his strains to the custody of WHO and their steering committee study group. I remember Charles Coburn being in charge of that at the moment from WHO, who took care of all the issues associated with the use of the thing, including what seeds, who gets seeds, what to do when the seed stocks run out, and so on and so forth. And one thing which was clearly lingering over this group was, are these strains from vaccine-associated cases actually vaccine strains or not? Okay, and at that time, it was not clear that they were. Okay, molecular biology had not been applied in any meaningful way to clinical virology at all. I don't think anywhere, really. I remember it first being applied to herpes, actually, but it wasn't really used there. So all the techniques that were being used to identify the strains were very, very classical. Okay, and what that meant for the vaccines was that you had a huge archive of strains and data. Nothing was ever thrown away, in my experience. There were histological blocks and slides and all the rest of it, and all the clinical data and so on, but nothing about the molecular biology of these viruses which were around. Okay, and finally, molecular biology actually came in to sort it out, and there were two main people that I would give credit to on that one. One was Eckhard Wimmer, who first published the oligonucleotide maps of different strains, type 1, type 2, and type 3, and showed they were very, very different, which meant potentially that you could tell strains apart. And the other was Vincent Racaniello, who did the first recovery of virus from a plasmid, which again, I think, opened the whole field to the possibility of doing real serious detailed genetics by modern methods. Okay, and for its worth, the vaccine was shown to cause poliomyelitis very clearly. Again, Albert Sabin didn't like it very much, but there we are. So by doing the mix and match experiments, looking at virulent strains and attenuated strains, what Jeff Armand and I did together was sort out a bit of type 3 here, which showed a determinant of attenuation in the 5' non-coding region at residue 472, which you've just heard about, and another one in VP3 at position 91, which was a phenylalanine in the Sabin strain. These two mutations, when inserted into Leon, had a very similar effect in terms of attenuating the Leon strain for monkeys. Okay, right. So they were equivalent, basically, for monkeys. Okay. So the next few slides are ones which I've nicked from Kostya, and thank you very much, Kostya, because they're much better than my slides, as you can see. MapRec was developed by Kostya for type 3. Okay. And the idea of this is that you produce a mismatch primer which introduces an extra cleavage site, so that when you do your PCR and you amplify that, if it's reverted, it cleaves. If it's not, it doesn't. And if you look at the picture down the bottom there, you can see where the cleaved product appears as shown by the arrow. Okay? And you measure the amount of cleaved product relative to uncleaved product, and that basically will tell you what proportion of reversion you've actually got there. Okay. I didn't like it. It was very clunky, I thought, but on the other hand, so was the monkey test, let's face it. So Kostya and I met at a meeting, and Kostya said, I can tell strains apart. I can tell strains which will pass the monkey neurovirulence test, and I can tell you what seeds they were made and what passage level they are. And I said, oh, yes, says you, basically, because I didn't believe him. Actually. So what he got from us then was about 90 different batches of vaccine, which had all been put into monkeys by the standardized WHO test, which was actually introduced in the early 80s, actually. And what you see here is if it's green, it passed. If it's red, it failed. Okay. And what you have along the bottom is the 472C percentage, and up the left-hand side, you have the mean lesion score, which is how these things are actually assessed. So what you can see is, I think, that MapRec is as good as the monkey test. Okay. In other words, MapRec, detecting just one particular mutation present in the Sabian vaccine strain, will do everything that a monkey will do for you in terms of these particular products. And this shows it even better, I think. So does that mean then that the SNP at 472 is responsible for the safety of the oral polio vaccine type 3? And the answer is no, it isn't. What it does show is consistency of production from one batch to the next. Okay. It only means that the only attenuating SNP that varies under production conditions and is measured by the monkey test or the MapRec test is at 472. And it could be possible to actually find production substrates that will actually keep 472 constant and revert the other mutation, but we don't use that. Okay, what we do is we use this thing. This is the only test which seems to reflect something about qualitative differences between vaccine batches. So basically, Kostya and I have finally come to an agreement, I think, that molecular methods will actually do, and particularly if you've got fancy molecular methods, they will do. Okay. This was a long and arduous road, and I'm really sorry I argued with you, Kostya, because you were right on this particular occasion. Okay. There's some points of disagreement still remain, you'll be surprised to hear. When comparing vaccine batches by NGS, HDS, MPS, other names, whatever you call it, it does seem to be a consensus in the field that you really only need to worry about SNPs which have a recognized biological effect. Okay. And I don't think I agree with that. I mean, I think if you've got a SNP that varies, then that shows lack of consistency of production, and you need to be sorting yourself out on that, I think. Okay. So any variation matters because it's a variation in your production process. Okay. And the other main point is you can control polio without eradicating it. And I really would like somebody to explain to me what that actually means. What is control and what is satisfactory, if you like. Okay. So what Kostya has done with the MapRec really was to introduce the molecular method which you can actually put in place to replace an extremely complicated and painful test. Okay. And maybe characterize your product rather better than you could even in the monkey, even though you're looking very, very hard. And while Kostya and I have bickered a lot over the years, I hope that we can actually continue bickering in the coming years. Okay, and I look forward to it. Thank you very much.
Great, thanks. It's a real pleasure to be here to honor Kostya. So I just wanted to give a little update on the Global Polio Eradication Program. And if you've seen any of the WHO maps lately, you notice that there's still quite a few dots on the maps. So this one in particular is 2022, showing wild polio in red, wild polio at AFP, wild polio environmental in pink. So you see that's up in Pakistan and Afghanistan, and then a little bit here in southern Africa. And then the kind of scary thing is that there's an increasing amount of CVDPV1 in this goldish color, so in a number of countries. And then also it continued high levels of CVDPV2, AFP and environmental, and then just a little bit of CVDPV3, which is typical of what we've seen in the past. However, one success is that wild polio type 1 in Pakistan and Afghanistan is at the level of genetic diversity that's lower than we've ever seen before. So there's a real opportunity. I know that that's been said before, but there's really essentially one genetic cluster in Pakistan and another in Afghanistan. It is true that the case numbers were low early in 2022, and then they increased. So far this year, there have been 2 cases in Afghanistan and 1 in Pakistan, and some environmental detection. So it is promising for Pakistan and Afghanistan. There was, as I mentioned, an outbreak later in the year in 2022, which reversed the trend of decreasing genetic diversity from 2020. So if you see this is a Bayesian skyline plot, it's just a way to give some idea of the genetic diversity over time. It is a lagging indicator, but you see this dip here in the low season, in the previous low season. And so hopeful that we're getting close there. Basically, most of the transmission is in South KP, starting from at least the beginning of 2021. And all the other genetic clusters that were present in 2020 have been eliminated from Pakistan. Of course, there's still caution. This is relatively short time frames of not observing those clusters, and cross-border risk exists for both of them. One challenge has been the importation of wild type 1 from Pakistan into Southern Africa. And so the first case was discovered in Malawi in November 2021, and then discovered several cases in Mozambique in 2022. And it's more likely that Mozambique had shared the virus with Malawi. It's impossible to know how long it had been in Southern Africa. There are some orphans in Pakistan, and so it's difficult to know, but most likely does not indicate a huge problem in that part of the world. However, it is very hard to keep population immunity up in so many countries, especially when polio starts to fall off the radar. So there have been difficulties in conducting high-quality SIAs in Mozambique, and also difficulties in coordinating the SIAs across countries. So as you know, the switch in 2016 removed the type 2 component from the OPV vaccine. And here we show 2016 from left to right, and then 2017. So this is a figure from May of 2017. So you can see the decrease in type 2 specimens, AFP and sewage over time. And then in a minute, I'll show you that then that increased, but it did initially look promising that what was planned was happening. However, Borno had VDPV in 2016 that had been missed for a very long time. So this particular emergence was at the time called CVDPV Chad A, and it had moved over to Nigeria. So if you look at this tree on the left-hand side, the timeline is 2012 through 2017 here on the right. And these red represent Nigeria, Borno from environmental and from AFP here. These long branches indicate orphan viruses where gaps in surveillance had missed those viruses for quite a while. The rest of the tree is generally Borno viruses, and you can get some idea of the likely emergence dates by working back towards the 7 that it emerged from. So this challenge then, detecting this VDPV after the switch, resulted in use of MOPV2, and then additional emergences were seeded at that point. So this is an extension of the diagram I showed before. This is post-switch CVDPV2 trend between 2016 and 2023, with AFP on the top, environmental in the middle, and then a collection of healthy children contacts, et cetera, on the bottom. And just showing from left to right, 2016 to 2023, this peak in 2020 of more and more cases of type 2 VDPV, and then some diminution over time, with 2022 improving somewhat from 2021. However, you see between 2016 and 2017, this increase that happened, 2019 coming up to a really high year in 2020, and of course, we all know what happened in 2020. So this has really been a challenge. It continues to be a challenge. This 2018-2019 period is where the cases started to come up. And then as responses were done with MOPV2, new outbreaks were seeded. So at this time point, there were more cases associated with VDPV than with wild, and that's true at this point also. More outbreaks than in the year prior to the switch or 2 years after the switch. More new outbreaks seeded by MOPV2 use than those that had been stopped. And at that point, there were 9 outbreaks and 29 outbreaks in the next time period after it, covering 17 countries, and then that increased over time. Some countries had multiple independent emergencies, which we've seen in the past. So in particular, DRC and Nigeria have done that kind of repeatedly. So just to show a little bit about the spread of the virus in that time period, a couple of years after the switch, 2 to 3 years after the switch. These are multiple emergencies on the right here. They range from emergencies from Central African Republic to Chad to Nigeria, and even one with Togo at the bottom here. In this yellow with red around it. You can see the viruses depicted by the different colors and them just moving from Nigeria or Central African Republic east and west. This is the year 2020, so you just see some more movement of the virus heading west towards Ivory Coast, Ghana, Burkina Faso, etc. For some of us who have seen this happen before with Wild 1 and Wild 3 in the past, and even with CVDPV, these are really concerning results. So to look at these outbreaks, we usually first do the VP1 standard report. All PV2s have to be sequenced in VP1 after the switch. When that rule was made, we thought that would be a relatively limited amount of time. However, we're still doing that in a number of sequencing labs around the world. In addition, we look at deep sequencing to look at the origin dates, sometimes better resolution we can get by complete capsid or the whole genome, depending on whether there's recombination or not. And then trying to look at the immunization campaigns that map to the emergence dates based on the molecular clock estimations. So some of these viruses fall into different scenarios, either pre-switch OPV2 origin, but outside of the time of MOPV2 use, or post-switch OPV2 origin around the time of MOPV2 use. Another category is post-switch OPV2 origin outside of that period of MOPV2 use, and now we have a complication of NOPV2 in addition. Just to give some idea of some of the emergencies in the 2018 to 2019 period in West Africa, this NIE JIS1 here on the left covered, oh dear, where did it go? Covered 13 Nigerian states, spread internationally, and was relatively divergent with 13 to 35 nucleotide differences from SABIN2. There were a number of emergencies around the same time in Sokoto, so they were numbered 3 through 6 and had 6 to 20 nucleotide differences. And then Kogi State also had one with relatively low, with 6 to 10 nucleotide differences, first detected in 2019. So this particular emergence that spread broadly, JIS1 from Jigawa, this tree shows 2018 to 2020 with 4 main groups of lineages that were spreading internationally. So you have the Niger and the Ivory Coast, then Cameroon and Chad in the green, Ghana, Benin, some of those that I showed in the map, and then even Burkina Faso, Ghana, CIV, Togo, etc. So really a lot of spread going on. And to look at the emergences, we often will look at other genomic markers. So this particular emergence has, as all of them do essentially, reversion of the attenuating sites. The 5' end was species C enterovirus sequence, and then multiple recombinant genomes indicating that either vaccine or species C had recombined. So this particular diagram looks into the estimate of the timing of the emergence of the origin date of the emergence, And in this one, once again with NIE-JIS1, the estimated date over here is roughly around September 2016, so just after the switch, with the range going from third, fourth month of 2015 to the end of 2016. So with that curve there in the middle, we can get some idea of the timing of when this OPV was given. And for one final diagram of this, this shows people here indicating the different immunization campaigns. So the bluish gray is bivalent OPV, the MOPV2 is in a goldish color, and then TOPV is in the purplish color. So from left to right, the switch is indicated by the red bar in April of 2016. And then downstream of there, the campaigns that map to this potential origin were right around September 2016 and between May and December. And in the states that had campaigns are indicated at the bottom. So in some cases, it can be difficult to know exactly which campaign might have started that particular emergence. So in terms of challenges, there have been many over time. I think COVID-19 was a big challenge in many ways, and you're all very aware of much of that. Resources have been pouring into the CVDPV outbreaks to stop them, and that's jeopardizing the ability to do preventative BOPV rounds. There's variable SIA quality, low quality immunization rounds in many places, and lower priority assigned to poliovirus in many countries, since polio has been gone from many of them for quite a while. Definitely widening gap in population immunity, especially to type 2 and type 1. And the complexity of vaccine choice just increases over time. There's a complicated vaccine supply landscape, and transition from MOPV and TOPV to NOPV2 has also complicated the situation. Definitely folks from the polio lab know that polio containment is a challenge, and in the GPLN, in the 146 labs around the world, they're working towards going towards direct detection in at least a subset of the labs. And so there's a pilot going on for that. That will introduce its own resource requirements and potential for cross-contamination and other technical challenges. And then from the lab perspective, there are increasing demands for sequencing. As I mentioned, VP1 sequence for all PV2s since the switch has been required, and that time period has been fairly long. Now we need more whole genome sequences for NOPV2 surveillance, and also in some cases to widen the window and get better resolution to understand the emergence, so looking outside of VP1. And that has been necessary for characterization of some outbreaks, such as the links between the US, the United Kingdom, Israel, and Canada. So initially, they only shared a couple of changes in VP1, and it was hard to link them. But with whole genome sequence, you could see the similarity in the genomes. Just would like to credit the labs in the Global Polio Laboratory Network in the 6 regions, and also credit the many partners. A lot of this work is funded by Bill and Melinda Gates, the CDC, WHO, and Gavi and UNICEF are really working hard on the immunization side, and then Rotary's been a firm partner from the very beginning. So I'd like to end just with saying what a wonderful time we've had collaborating with Kostya. Before I got to the lab, which was a while ago, there was already a long history of collaboration, and we would turn to his papers for methodology for long PCR and deep sequencing, for example. He had some of the first expertise in deep sequencing. And as I was looking back, I was reminded that he did the early transgenic mouse neurovirulence testing of some of our codon de-optimized viruses. We were excited to invite him to join us as a partner in the NOPV2 consortium, and he did really expert work on deep sequencing, and Olen's going to say a little bit more about that in his talk in a little bit. So congratulations, Kostya, and it's been really a pleasure working with you. So just one more thing, which is photos of the consortium, the NOPV2 consortium in Napa in 2017. So Kostya is over here on the side, and we're enjoying the beautiful scenery. and then a couple with Ellie, Kostya, Stan Lemon, Raul, and Andy. So would like to just acknowledge a lot of people involved in both at CDC and the other labs, and pause. Thank you so much.
First of all, I want to thank Amy and George for organizing this beautiful symposium in honor of Kostya, and to invite me to join with the reunion of Kostya's friends from all over the world. And it's a real honor to be here. As you've already heard, there is something which Walt Dowdle called the OPV paradox. And before he coined that term, at the ICV in 1990, Vadim stood up in the audience and asked me a question after I had given a presentation about the progress in the Americas, which was nearly complete by that time. They eradicated polio from the Americas by 1991. And Vadim's comment was, well, this is all very, very encouraging, but how do you stop with the use of OPV? So as you've already heard, the OPV paradox really is the eradication of the wild poliovirus. For example, wild poliovirus 2 in 1999. You no longer have natural immunity, so there's widening immunity gaps in low coverage areas, which unfortunately we have many of in the world. There's then uncontrolled spread of excreted OPV derivatives to successive contacts, full recovery of the wild poliovirus phenotype, and increasing, as you heard from Cara, CBDPV2 outbreaks after 2000, and also BDPV1 and BDPV3. BDPV2 is by far the most common, BDPV1 next, and then CBDPV3, least common, a little bit counterintuitive of what we might have thought from Phil Minor's observations and those of others about vaccine-associated paralytic polio. But type 2 is clearly the one that has caused the greatest problems, as you've heard. So the WHO tried the conventional approach, and as you'd already heard, the plan was synchronized global withdrawal of OPV2. And they chose the lowest point of the polio season in the Northern Hemisphere to do the withdrawal, to stop using the Sabin type 2 OPV component. And ahead of that, they tried to close the PV2 immunity gaps with IPV. But of course, the problem with IPV is it does not cause intestinal mucosal immunity. So it causes humoral immunity, blocks respiratory transmission, but does not block fecal oral transmission. The idea was to use Sabin OPV for outbreak control if need be. The plan, as Cara had told you, was quite comprehensive, with withdrawal of all OPV lots worldwide, all TOPV, with very detailed documentation of their destruction. But this is a worldwide activity. In the parts of the world where they still were using trivalent OPV, and there were performance gaps. The main performance gap was, as you'd heard, that there was still circulating BDPVs, particularly in Nigeria and elsewhere, but particularly in Nigeria, that were undetected. There are also gaps in polio vaccine coverage. There was poor coverage with IPV in many areas, and there is a rising tide of vaccine refusals in many of the highest risk areas of the world. So as you'd heard, there was an upsurge of CBDPV2 outbreaks after 2016, and the use of Sabin OPV in the underperforming areas sparked new outbreaks, which is our greatest concern. So this highlighted the need for a genetically stable OPV2. Now, another solution would have been to maintain and obtain high levels of polio vaccine coverage worldwide. But that's been the goal since 1985 when PAHO launched its program, and it's still not been achieved in certain key parts of the world. So this need for a genetically stable technical solution to this problem was anticipated in 2011 by the Gates Foundation. and they supported the establishment of a new OPV2 consortium. Now, the consortium included 3 research teams. One, you've heard from already, was the NIBSC UCSF team of Andy McAdam and Raul Andino. The other was the CDC FDA team with Cara Burns taking the lead from CDC and Kostya Chumakov from FDA. Then Stony Brook University and Eckhard Wimmer with his contributions also were entered into the consortium. Now, we immediately asked Kostya for help. So the Gates Foundation initially requested that we join the consortium, and we invited Kostya to join us as a partner. And we were hoping very much that he would accept our invitation, and we were very delighted when he did, because he brought many key areas of expertise in vaccines, vaccine characterization, interpretation of high throughput sequencing, tremendous amount of experience to this collaboration. And we've enjoyed his work with us the whole time. But in addition, he did the work in a timely way, and it was very, very high quality. So one of Vadim's star students would help address the incisive question that Vadim had posed 3 decades earlier. 3 decades earlier. But there's also a kind of a symmetry here, because Kostya's father, Mikhail, had helped start OPV use, and Vadim was now going to help stop OPV use. So our approach was codon deoptimization across the capsid region. And the idea was to replace preferred codons with synonymous unpreferred codons. Now, this codon deoptimization is reducible to the desuppression of CpG and UPA dinucleotides, which Vincent Racaniello and Eckhard Wimmer had shown in 1981 was naturally suppressed in poliovirus genome. So already the hint was out there. And the hint was not only were there preferred codons, but there were unpreferred dinucleotides, particularly CpG and UPA. So we introduced more than 100 substitutions, each of which incrementally reduced the infectivity of the virus. That would predict very high genetic stabilities, and we would infer there'd be very low selective pressure on each engineered change, and that was confirmed experimentally. We referred to it as very low individual selection coefficients for change, and Eckhard, who took a very similar approach, described it as death by 1,000 cuts. There would be an unaltered amino acid sequence, and there would be a prediction of an unaltered antigenicity, which our consortium partners confirmed experimentally. In addition, codon deoptimization in polio in the capsid conferred temperature sensitivity to the virus. And temperature sensitivity is a favorable property that is common in many attenuated vaccines. So this is the figure 1 from Jennifer Kanopka-Onstadt's paper describing the codon deoptimization application to type 2. And we started off with MEF1, which is the wild type 1. That's actually the IPV seed strain, grows quite robustly. And you can see as we increase the amount of CpG substitutions right up to saturation, this is CpG 2-3 position, the plaques grow smaller, and indeed in single-step growth curves, the virus grows progressively more slowly as the amount of CpG is introduced. And this is a plaque area measurement, this curve, with MEF1 at 20%, 40%, 60%, 80%, and 100% saturation just in the capsid region. And this is Sabin2, which is already having a smaller plaque area. And this is our candidate, NOPV2. It became candidate 2 in the Gates Foundation consortium. but in this paper it's described as NOPV2 CD, codon deoptimized. There is a nearly linear fitness decline to increase CpG substitutions. So that was an advantage because the replicative fitness of the virus can be fine-tuned. And you could, as I think Cara sometimes said, you sort of dial in the amount of fitness reduction you want. And UPA desuppression has similar effects. The slope of the curve, dose response curve, is more shallow for UPA than it is for CpG. But UPA and CpG can be independently modulated as a strategy. So what we were trying to do was to mitigate the consequences of recombination, because we'd seen many cBDPVs from all over the world that had double recombinants. They had non-capsid protein sequences derived from species C enteroviruses, including other polioviruses, vaccine strains. And the 5' UTR also had recombination. So this is the left recombination zone, the right recombination zone. The exchanges are more frequent on the right side and the non-capsid side than it was on the left side. But that's perhaps in part because this is just a smaller area for crossovers to occur. There's a no recombination zone here where we saw no cross-serotype exchanges, but exchanges within a serotype were still frequent in natural infections. So the key point is recombination accompanies poliovirus replication. Most cBDPVs are vaccine, non-vaccine recombinants, and many retain only the poliovirus capsid region sequences. So our thought was, what if we replace the capsid region sequences with the deoptimized codons, or in this case, desuppressed CpG. So what we did is added 109 additional nucleotide changes. That would be called CpG 2, 3, 40%. And adding to that was Andy McAdams' Sabin S15 domain 5, which Raul has already described, which has greatly stabilized the attenuated phenotype as tested in experimental animals. So that was our construct. And it basically, this is the area that is not exchanged across serotypes. So one of the things that had to be determined was the genetic stability of these constructs upon passage in cells. Later, the clinical trials would test the stability in children. But just to get started, we had to look at the accumulation of mutations in serial passage in Vero cells. And Vero cells are selected for reversion of the 481 position and for other changes. And so this was after 10 passages, a rigorous test for mutation for the genetic stability of the candidate strains. So this is Sabin OPV2, and there's a number of changes that are occurring in Sabin OPV2 as detected by Kostya by deep sequencing. And then this is our candidate OPV2, and it was much more stable. And this change here, which is seen both in Sabin 2 and in the R candidate, turned out to have rather no evidence of any adverse role for this phenotypically. Now, we were aided not only by Kostya, but by the collaborating team members of Majid, Tatiana, Svetlana, Eugenia, and Diana, all of whom contributed to this collaboration, which was a very, very productive collaboration. So Kostya's conclusions were that candidate 2 is significantly more genetically stable than Sabin 2. Neurovenous mutations accumulate very slowly, and there's no evidence for an adverse role of this one prominent change. So these findings, along with the other findings, but these findings from Kostya helped set the stage for the clinical trials of Candidate 2, sponsored by the Gates Foundation. Now, I don't want to go into the details of the design of the Candidate 2 beyond what I've told you, but it's published in a paper with a number of collaborators, including Kostya Chumakov, who played a key role in its development, along with Raul and Andy. But I do want to just highlight some of the conclusions from 2 clinical trials, because the 2 vaccine candidates, that's 2 novel type 2 oral polio vaccine candidates, the 2 candidates were tested largely in parallel. And the conclusions were that both novel OPV2 candidates were safe, well tolerated, and immunogenic in children and infants. That was one conclusion. And then in a second study, both novel OPV2 candidates showed superior genetic and phenotypic stability compared to Sabin2 after replication in children. So the rest of the story is that this is a product of active collaboration between NIBSC, UCSF, and the CDC FDA teams in characterizing both candidates leading up to the clinical trials. Now, the Gates Foundation sponsored clinical trials and subsequent laboratory analyses brought in many more collaborators encompassing a wide range of expertise, and their names are in the previous slide. Candidate 1, which had no capsid modifications developed by the NIBSC UCSF team, was ultimately selected for production. 600 million doses have been distributed. Its Candidate 1 is now called novel OPV2 and has performed well in the field for outbreak control. And it's unfortunately for Albert Sabin, is proven far superior to Sabin OPV2 in the field. And that's both from what Raul's told you and from what Paul had told you earlier. So on this special day, I want to repeat what's been said by many. Congratulations, Kostya, on your stellar career. I want to thank you for your highly productive collaboration with us. Also, I want to convey the thanks from the Bill and Melinda Gates Foundation for your contribution to the consortium. And I think, as Vadim has already very, very eminently demonstrated, as you approach retirement, you will have the reminder that good science and good scientists never go out of style. So I wish you a long and happy and well-earned retirement. Thank you very much.
So, yeah, it's really an honor. And thank you very much, Amy and George, for the invitation and the possibility to talk here. It's really not only, important meeting because we are celebrating Kostya's life, scientific life, but also because it represents, really all the polio virologists and also those that don't work in polio coming together to celebrate his work. And my presentation obviously has to do with this idea that polio still is around and is not dead. And I wanted to talk about evolution. This is where I put Darwin, but really it should be replaced by Kostya. And it's been, to me, an honor to know Kostya and all his friends coming from Vadim's lab. Over the years, we really developed not only scientific, but also very close friendships. So what I wanted to just share is something that many of you know, but I just wanted to present a sort of an overview. This is the picture of the last 22, 24 years. 1988 to 2020. Clearly, the saving vaccine worked. It's easy to produce, transport, delivery, no needles, no doctors needed for this vaccination campaign, but induce good mucosal immunity and prevent transmission. And also, it's a transmissible vaccine, which increased the coverage of the vaccination by from vaccinee to contact individuals. And so it's been very successful. But of course, if you don't have a complete vaccination, a good coverage vaccination in the population, this enables viruses that are shed from the vaccinees to circulate in the population. And over time, this thing can develop viruses that are regain fitness, circulation capacity, and virulence. This is a picture of the 2020-21 situation. Many of you have seen this. This probably was exacerbated by the SARS-CoV-2 pandemic, but you see all these dots in green represent circulating vaccine-derived polioviruses from type 2. But also you can see that type 3 and type 1 also could emerge from vaccination. Not only that, but closer to home, new cases of either a polio outbreak in New York, London, and Jerusalem was detected. So these viruses do not circulate only in these areas where you can absolutely see manifestations of the infection, but also can easily be circulating in Europe, United States, and other places. So we thought several years ago that it was important to try to understand how these viruses get de-attenuated, how they became virulent. And it was an opportunity to study this in this parallel evolution. You started with this saving strains and over passages. You end up with these viruses that cause paralysis or can cause paralysis. And we collaborate with Adi Stern, David Ye, and Andy McAdam at that time to try to understand, using maybe some statistical new tools, a Marcos model approach that we call Paracel, to try to identify what substitutions were important in terms of making the saving more virulent. And it turns out that only, based on this analysis, only 3 mutations were required, 2 in the 5' UTR and 1 in the capsid protein. This was not completely surprising. This has been reported previously, but using these new tools, we sort of were able to confirm these observations and extended a little bit these studies by doing work in both cell culture and animal models and reconstruction of some of these viruses that were observed in the field. Now, one of the other things that was observed in this study is that there was a lot of recombination with another class C enteroviruses that co-circulate with during vaccine campaigns. And you can see here, this is similarity of the genomes with respect to the original saving vaccine. And you can see that there's some regions here where the similarity is much reduced, both in the 5' UTR as well on the non-structural proteins. In this particular study, we also examine samples derived from individuals who were vaccinated with the trivalent saving vaccine, and samples were collected 14 days after, and studied by next generation sequencing. And what was obvious is that these 3 mutations, the 2 and the 5' UTR, this mutation 481 was almost fixed in all the individuals that we analyzed. In addition, we observed, since this was a trivalent OPV, that many of the individuals had also recombinants between type 1 and type 3, or type 2 and type 1. So based on this sort of analysis and analysis from previous analysis as well, we hooked up together with Andy McAdam to try to see whether we could try to slow down the evolution of the virus. And so there's 3 steps to understand what is the basis, the molecular basis of this, more stable, genetically stable strains, the new, what called new OPVs. And one of the elements is this element that is here in 2-dimension representation. Really, we don't know the exact structure of this element, but what is clear is this position 481 here is an A, and when mutation, the A is mutated to a G. The G seems to be able to, at least the prediction was it could interact with this C here, increasing the extent and the stability of this stem loop, and increasing the overall stability, physical stability or thermodynamic stability of the structure. And this was confirmed or further supported because there were additional mutations observed. This was the most common mutation observed, but there was additional mutations in other parts of the structure, which were GU base pairs, no Watson and Crick base pairs, that were converted to GC base pairs, more stable. And the overall delta G, the free energy of stability of the structure, was increased by this single point mutation, these simple point mutations. So the idea really was pioneered by Andy McAdam in Phil Minor's lab, and also Jeff Almond. And I was trying to convert to work with the structure to maintain the delta G, the structural stability of this RNA structure, similar to the saving, preventing that single point mutations can actually increase the stability of the structure. And it was amazing that there was a strict correlation between delta G and neurovirulence, both tested in cell culture, but also in animal models. So that was the original. They did a very empirical series of mutations, introduced mutations here to come up to a structure called S15, which were now used in this development of these new strains. And the idea was that this would preserve the physical stability of delta G, preventing that few single point mutations could increase the delta G. But of course, as I mentioned before, some of the recombinants, the revertants that you see in the circulating, they have a recombination in the 5' UTR. And so the second modification of these viruses or the saving strains was to deal with recombination. And as you know, recombination by copy choice can replace large fragments of the genome, eliminating some structures. So it was easy to imagine that this structure could actually be replaced. The more stable structure could be more replaced by structures obtained from co-circulating viruses. And so to prevent that, we focus on this other structure, the CRE, the cis-actin replication element, and critical element for the initiation of RNA synthesis in picornaviruses. And the idea was to eliminate this structure or mutate the structure so it's not functional anymore in this position, and move the wild-type structure closer to the structure that we want to maintain in the genome. In this case, by having these two structures together, a recombinant will have to reconstruct this structure here, the CRE element here, and also try to replace the 5' UTR, the whole 5' UTR, with another co-circulated enterovirus. And that required two independent events of recombination. And we thought that also was something that could help to prevent this simple reversion by recombination. And the third thing is that, as you know, the RNA-dependent RNA polymerase in poliovirus is able to introduce mutations, and it's also the enzyme that is in charge of recombination. And so we using some genetics approaches, also the Julie Pfeiffer and Kirker's lab, obtain mutations in the polymerase by passing the virus in the presence of mutagens, and also recombination could be selected also in a genetic screen by using this recombinant EGFP. And both of this, a combination of these two defects, the increasing fidelity and decrease in recombination, also reduce the ability of the virus to adapt, and also attenuate the virus in animal models. So the combination of these three approaches, the Cree element, domain 5 with all these mutations, mutation in the Cree, and a combination of high 5 and recombination, yield this virus that we call NOPV2. And this was previously published. And each of these modifications, importantly, contribute to attenuation and stability of the virus. Now, it was a big question because this virus was actually attenuated and immunogenic in preclinical models like in mice, but the big question was whether this would work in humans. And so phase 1 clinical trial was conducted where few patients were given oral vaccination with this new vaccine, and virus was studied and also observed neutralizing antibody, increase in neutralizing antibody titers. And this graphic was actually very, very reassuring, and we were very happy when we saw that this virus was able to take in this in humans by oral inoculation. And actually the pattern of replication and clearance was very similar to that of saving. After that, from this phase 1, based on this studies and also with the analysis of sequencing showing that the virus was stable in the more genetically stable in these conditions, a phase 2 clinical trial was conducted in Panama, where now there was a comparison between the new OPV with the original saving type 2 called MOPV. And you can see that if you compare MOPV2, there is many individuals sharing large proportion of viruses which contain the mutation in 481 and a few other mutations, whereas this is not observed in NOPV2. Perhaps more importantly, functional analysis of viruses isolated from these patients were examined by spinal inoculation of susceptible mice and looking at paralysis 14 days later. And you can see that at this concentration for logs fused, many animals were paralyzed if they were vaccinated with MLPV2, but none of them by NOPV2. You increase the doses, you start to see some animals paralyzed with NOPV2 as well, but there's still very significant difference. And so based on this, there was clearly evidence that the vaccine was stable, but also immunogenic, and it was also clear that the vaccine could be an improvement compared to the saving. And so WHO approved this vaccine, and since then, many millions of doses in many countries have been introduced, demonstrating the efficacy and the stability of this new vaccine, including to stop CVD-PV circulation. More recently, of course, you have other type 1 and type 3 from saving, and we've been developing also more stable NOPVs with type 1 and type 3 capsids. And the strategy there was a simple construction of chimeric virus, where the saving capsid of 2 was replaced by saving 1 and saving 3 capsid proteins, but the rest of the structures where all the modifications were located were identical to the original saving 2. And we had the advantage at this point that a lot of the clinical studies yield information on what can be the type of mutations and sequences changes during those vaccination with the new NOPV2. And so we introduced many of these mutations to try to identify whether the new NOPV1 and 3 were also genetically stable, even if they incorporate some of the mutations observed in NOPV2. And so you can see many mutations were located in the 5' UTR, some in the capsid proteins. And so if I can walk you here, these two lines here, what we're looking here is PD50s and susceptible mice, and here's saving 3, which you need about 3 logs to paralyze the animals, and the PD50 is about 3 logs. With Leon, it's about less than 1 log. So this is the difference between the vaccine and the wild-type virus. If you introduce a single point mutation in domain 5 and saving 3, now the PD50 is reduced to half of saving. The new OPV3 construct is much more attenuated. But the important thing is when you introduce all these mutations in this strain, the attenuation still is maintained, and it's only when you replace you put all these mutations and also replace the structural proteins by Leon proteins that sequence, that now you get to a point that you have similar neurovirulence that compare very close to the original saving 3. So even with all these mutations, this strain, this potential strain, worst case scenario, still looks like very much like original saving. So this is immunogenic, but this opened the opportunity to now develop trivalent formulations. So this is a comparison of trivalent saving with trivalent new OPV. You can see that it's comparable immunogenic, and you can protect many of the animals with the trivalent OPV too. So this is not only that produce good neutralizing antibodies, but also that is immunogenic. I was hoping, I just wanted to mention that in very recent time, in March 2023, WHO reported that in the Democratic Republic of Congo and Burundi, there were 2 separated, both in DRC, emergence of strains that cause paralysis, unfortunately, in kids, and they were derived from NOPV2. And I wanted to just mention that saving originally was effective to halt spread of wild-type polio and eradicating most of the polio globally if high-quality campaigns were carried on. Of course, there was a problem of vaccine-associated paralysis, but if you don't vaccinate sufficient numbers of people in the population, then you also have circulating VDPs. NOPV decrease the probability of VAVP and also CVDPs. I think there's enough data to demonstrate that. But the situation in Burundi is important because it shows what are the limitations of this improvement in the medical intervention. So, and it's important that using this information, we can further design campaigns that will prevent these unfortunate cases. Injectable vaccines probably will not replace NOPV because the level of coverage of oral vaccine is much higher, but also because the injectable vaccines in general are not very effective to prevent the spread of viruses. This has been not only the development of these new vaccines has been the product of the entire field and the experience and the information collected in basics research in antivirus in general. And so it's really great to have a field that has been so productive and so friendly for everybody. And the only thing I wanted to just mention that improving vaccine is maybe not very glamorous, but very important, because as any medical intervention, you wanted to keep improving these things that save life. So here's a sort of group of pictures collected over the years. You can see Amy Weiner and David and Andy. There was a lot of constructions going on in this project. Ellie and Olen and Jen and Cara and Kostya, Ananda and John Carnes. And of course, Erika at the beginning was working with us. And here is interesting. Obviously, Bill and Melinda Gates Foundation supported this study, and he's saying, we are really close to ending the polio, ending polio. But this is an old picture. If you look at a more recent picture, you see he looks a little bit older, and his fingers are a little closer. So I think he thinks that we are closer now to end polio. Perhaps using these new tools and develop in the context of this consortium. That's it. Thank you very much.